Hi all, this is Group 4. We are going to present a treatment of COVID-2000X. Here are the contents. Introduction, Solution 1 about existing drugs, Solution 2 about new therapeutic strategy, Conclusion. Introduction. Controlling and managing an outbreak is one of the most difficult tasks of public health. Moreover, uncontained outbreaks can easily lead to local epidemics at best and global pandemic at worst. COVID-19 is a prime example of global pandemic which originated from a such outbreak in Wuhan, China. The purpose of this presentation is to describe treatment options in the event of hypothetical outbreak of a novel virus COVID-2000X. We have undergone diseases associated with coronavirus, 2002 SARS, 2012 MERS, 2019 COVID-19. And what will be COVID-2000X? We will follow the assumption that the virus will be in close genetic relation, have similar pathogenesis and pathophysiology to COVID-19. We'll be solely focusing on curative therapy and ignoring symptomatic or supporting therapeutic strategies.
Okay, then I'm gonna talk about the treatment using the existing drugs. We can use RNA-dependent RNA polymerase inhibitors. And the first example is remdesivir. It targets viral RNA-dependent RNA polymerase. Since remdesivir shows great effect on pulmonary load in human and zoonotic coughs, it is the most promising antiviral treatment for COVID-202X. Verviparavir also stops RNA polymerase of coronavirus. By the way, this drug is still under study because of its side effects like fetal toxicity and death. And the other way is to use protease inhibitors like lopinavir and ritonavir. The drug contains these two antiviral components for blocking virus replication. Hydroxyl group interacts with the carboxyl group of the protease active site residues and deactivate them. Actually, it makes the positive effect for late stage of SARS-CoV-2. And chloroquine or hydroxychloroquine can be utilized for treatment of COVID-202X. They prevent virus to cell fusion by increasing endosomal pH level and interfere with glycosylation of the cell receptor. Hydroxychloroquine is the one with lower the toxicity and potential for drug-to-drug interaction than chloroquine. Both of them are promising broad-spectrum antiviral drugs for COVID-202X.
Hi, now let's talk about proposal of a new therapeutic strategy against coronaviruses. Angiotensin converting enzyme 2 is an enzyme that is located on the surface of your cell. And most coronaviruses, such as SARS and COVID-19, use this receptor to enter into your cell. The key advantage of targeting ACE2 is when patients are administered a drug that would bind with ACE2, the target in this case is the host, not the virus. Viruses are capable of rapidly mutating in order to be resistant to the therapeutic agent. However, if viruses mutate, the entry receptor into the cell is usually reserved. That means no matter how many times a virus is mutated, it will still use receptor ACE2 to enter the cells. That's why targeting ACE2 receptors is a very promising therapeutic strategy. Right now, there are three agents that can disrupt virus to ACE2 binding. First is smart receptor binding domain, or RBD. Researchers have created RBD from spike protein or S protein. When we add RBD, RBD binds to ACE2, and thus ACE2 is blocked and a virus cannot enter the cell. RBD is very small in size. That means it can it can perfuse into tissues very effectively. It's cheap to produce. And because RBD and S proteins are practically the same, have the same structure, uh, if RBD is perceived as an antigen, it leads to antibodies that would also destroy coronavirus, which is called the vaccination effect. The second option is single chain variable fragment, or SCFV. This fragment was created from a ACE2 specific antibody, and this fragment also can bind with ACE2 receptor and thus blocking a virus entering the cell. So it's even smaller than RBD, and because it doesn't have any FC domains, there will be no immune response against this protein. The third option is ACE2 FC fusion protein. Now, this fusion protein is a little bit different because RBD and SCFV would block ACE2 receptors, but this fusion protein already contains ACE2 and FC domain of an antibody. So this protein binds with S proteins of the virus and blocking it to going into the cell. And because this protein has FC domains, there's gonna be an antiviral immune response. And this protein is much cheaper to produce than monoclonal and polyclonal antibodies. Now, these are three options that can target ACE2.
And here are the conclusions. We can predict future COVID-2000X by paying attention to the characteristics of the coronavirus. Suggestions using existing drugs are RNA-dependent RNA polymerase inhibitors and protease inhibitors. Global suggestion is using ACE2 receptor as a therapeutic target. And these are references. Thank you, and any questions?